for your patience as we worked out some technical difficulties. So today we have Dr. Adam Chicco joining us. And so Dr. Chicco is an associate professor in the Department of Biomedical Sciences here at Colorado State University. He received his Ph.D. from UNC in Greeley and then performed a postdoc fellowship with Dr. Russell Moore down at UC Boulder. Dr. Chicco joined CSU back in 2008 and actually started in our department and then moved over to biomedical sciences. Dr. Chicco has really published quite a lot in his time as a faculty and as a trainee, over 50 papers, really authoritative in the fields of cardiovascular physiology and mitochondrial energetics. But what I'm excited for is today he's going to talk about results from an even more interesting tissue, in my opinion, which is skeletal muscle and its adaptations to altitude hypoxia. So thank you, Dr. Chicco, for joining us. I'll let you take it away.
Thanks, Dan. And thanks, Tom, both for the invitation. It's great to, I think this might be the first time I've given a talk since I moved over to BMS. And yeah, I'm hoping this will be of a real interest. This was actually a study, a lot of the data I'll present will be from a study that was done while actually I was still over in HES. It's been a long time. 2012 was when the tissues were collected, and it took obviously a long time. I think we published the paper in 2018, but anyway, many people that who I think are on the call know about this study and have been helpful even in discussing some of this data, I think, over the years. You'll see pictures of a couple of them. I'd be lying if I said that I was interested in high altitude hypoxia because of the insights it can provide to hypoxia-related conditions, though I've said that many times in my career and printed it. And it's true to some extent. But if we're honest here, I've always loved the mountains. This is a shot of me up on Humboldt Peak down in the Crestones here in Colorado with the Crestone 214ers behind me. And so I've been fascinated by mountains and high altitude activity for many, many years, as I know a lot of people in HES have. One of the big draws of Colorado. So it was a real pleasure to be involved in research to understand how muscle responds, and I'll get to that. If I can advance, yeah, okay. So just by way of a little overview, I'll talk briefly, and I hope the students in the audience will see maybe even some slides that they've seen before. I used to teach HES 403 and stole a couple of my slides, modified now a little, on the physiological responses to high altitudes. So I'll do a brief overview there, and then get right to the bioenergetics, muscle bioenergetics piece, which has really been interesting, an interesting journey. I came into it pretty late in my career, again, more as a side project, but I'll give an overview of the current perspectives and controversies in the field that are still pretty hot. Not that people are arguing about it, but yeah, it's not really clear exactly what's going on, and I'll kind of explain why. And I hope that my study is helping to really at least nail things down, or at least show how everybody may be right and wrong at the same time. And hopefully that'll make some sense. Specifically, I'll go over some of the data we have from the paper that I think Tom sent out that was published in 2018, but it's a pretty dense paper if you looked at it. And I'll try to do a better job explaining it here in person or virtually than I could in cramming it into a manuscript. But this was an interesting multinational scientific expedition down in Bolivia that was done in 2012, led by Rob Roach down in Boulder, Denver. Actually, he came up and gave a seminar at HES, and that's how I met him and led to my involvement with that study. I'll try to finish up, hopefully not too much later than 4:30. Just kidding, but hopefully not too much later than 4 or quarter till, with some take-home messages that I hope I can convince you is one thing we should always, or a few that we should really think of when we're studying altitude muscle adaptation in general. I'll also do a little bit on our current directions. We're actually in the middle of a study right now on this that I'll talk a little bit about. Okay, so obviously it's stressful at high altitude. These three guys here, one of them, here's Dr. Hickey and Chris Melby leading the way up the Dragon's Tail Couloir in Rocky Mountain National Park, a favorite regular spot for us. I can't remember who this is, I think a PhD student. But obviously, when you're at high altitude, there are many things that make the activity more difficult, even if it isn't steep snow like this. And the big one is the low ambient PO2. Of course, the fraction of O2 in the air is still 21%, like it is everywhere on Earth, but the barometric pressure here is lower as you go up in altitude, and so the partial pressure of O2 in the ambient air is less, and you ultimately breathe in fewer oxygen molecules per breath, which drops the O2 in the alveolus and so forth. It's also cold often, although not always, and very dry, so dehydration can be an issue. And then, as I'll talk about, you get a lot of sympathoadrenal drive that disturbs your ability to sleep, feeding cycles, and all of this is made a whole lot worse by the fact that most people who are up here doing things are physically active. And that, I think, has been one of the big controversies or source of controversy in the field is, we're asking these questions, we're often doing it in subjects that are, been opportunistically recruited to be part of studies when they're really interested in climbing mountains or trekking, where obviously there's a lot of stress associated with that work. So these are just graphs I think undergrads have definitely seen as the altitude increases, the ambient O2 and then the alveolar O2 drops. VO2 max, our maximal capacity for the body to consume oxygen, pick up those electrons in the mitochondria to support ATP production, also declines with increasing altitude. So it's harder to do the work the higher we go. And of course, this is another one I think we're all familiar with from Brooks, at least a really early version of the exercise phys text that he put out. And the oxygen transport cascade essentially shows this gradient of PO2 that we have from ambient air at sea level here, and then all the way down to what it could be inside muscle mitochondria, or the myoglobin that would kind of hand off the O2s to the mitochondria inside muscle cells. And it's a pretty steep gradient. But of course, at altitude, that gradient is lessened because there's less O2 partial pressure, less O2s wind up in the alveolus, and less O2s wind up in your blood. And so high altitude induces hypoxemia. And that's a really important word when we talk about what we know is happening at altitude. Hypoxia could refer to the air, could refer to tissues. But hypoxemia is what's going on in the blood. And that absolutely happens. We can see it as a hemoglobin desaturation. We can see it as low arterial O2 content. And whether or not the cells that the blood is going to are hypoxic is another question. And I'll kind of get to that. Of course, we have a wonderful series of steps here that we can have adaptations to improve with regard to pulmonary blood flow, peripheral blood flow, oxyhemoglobin binding, capillary density over time can change. Access to different tissue beds obviously can change with vasomotor control, and even down at the tissue level, our ability to release O2 from hemoglobin is can be altered by blood pH and all these things that we know happen during exercise and even just in altitude. So I'm not going to go over all of those, but the point is at the bottom of this long cascade is the muscle cell. And in there, we have a beautiful level of complexity as well. But much of what's going to happen down in here is going to be dependent on how well these adaptations above or processes above have succeeded in supplying the oxygen. And so I'm going to focus all of this talk, all of it really, on muscle mitochondria and metabolism. But it's important to remember that the adaptations upstream are probably more impactful in terms of keeping you able to function and maybe even adapting to high altitude. And so the muscle is probably responding largely to picking up the slack, if you will, to whatever isn't happening upstream. But there's more to it than that as well. Even if the oxygen delivery is there's more to it if the muscle has enough oxygen to what happens at high altitude. And I'll get to that. But in the cell, the ATP levels, or ATP to ADP ratio, is really what drives how much metabolism or energy metabolism is going to be occurring. And it does that, of course, by if ATP drops, it's going to pull electrons through the ETC and power the ATP synthase with the proton gradient that ends up pumping. So it drives metabolic demand. It also influences, as you might recall, some of these key rate limiting steps in the oxidative pathways as a means to kind of regulate upstream flow of fuel down into this, this battery. So the ATP-ADP ratio is a really important regulator of metabolism, and I'll come back to that. Both by creating the demand and also allosterically regulating key enzymes that regulate those processes. Oxygen, of course, is the final electron acceptor. If you remember, it comes and picks up these electrons that you're plucking off of all of these substrates on the way down, and they're essentially used to power these pumps that will generate a proton gradient to fuel the ATP synthase. You also can, of course, make just some ATP from glycolysis alone, right? With no oxygen needed to pick up electrons, we just have a lactate produced as a byproduct, and if that accumulates, it can be ejected from the cell along with a proton, which of course drops the pH in the interstitial space and ultimately the serum if it gets high enough, and that can be sensed by the central nervous system, feedback to regulate all kinds of things, all of these functions actually. The liver can take the lactate, turn it back into glucose. There's the Cori cycle, and when you start having any neuroendocrine control of stress, if you will, when this is happening, or acidity triggering things like sympathetic drive, you're going to have metabolic responses too that feed into these pathways. Hypoxemia itself does lead to this sympathoadrenal activation. So epinephrine or your catecholamines and cortisol are elevated when you have this response, or when you have this exposure to high altitude or whatever might be causing hypoxemia, and that can drive a lot of this metabolic remodeling, if you will, over time. And that's really our hypothesis now that I'll try to get to today. One of the things that I haven't mentioned yet that is occurring when cortisol is high, glucocorticoids are going to trigger muscle catabolism, so proteolysis. And this creates a problem for muscle. Even if you have plenty of muscle to contract, or myofibrils are still working, you have an accumulation of amino acids. This is something that the muscle cell has to deal with. We can't just have a whole bunch of excess nitrogen around, and you'll see that it kind of rewires metabolism. And what our data suggests is that that's actually a big driver of a lot of the changes you see in the muscle. And it's not even due to hypoxia, perhaps. It's simply the cortisol or sympathoadrenal drive that drives all this fuel mobilization, including amino acids, that the muscle now has to handle. So the question is, does the skeletal muscle adaptations or metabolism contribute to performance and in order to, or improvements? If you're up there for 2 weeks, we all know that things get a little better. There are all kinds of adaptations upstream of the muscles that are well described. So the question is, are the changes that are happening in the muscle, are these good? Are they bad? Are they contributing to the acclimatization response? That is the improved performance and capacity for work that we have up at altitude over time. And in order to know whether it's good or bad, we really have to understand exactly what it is. And so that's been really controversial. And largely, I think it's because the amount of work that's been done is actually pretty extensive, but it's all over the place. And it's fueled by, I have apples, oranges, and love the mountains. Many, many, many studies that are just not comparable. Even human studies at high altitude won't be comparable. Some are climbers, some are not climbers, some are at 4,000 meters, some, climbed Everest and came down after. And you can't blame these folks because, a lot of this interest was driven simply by the love of going to these places and doing these kind of cool field studies. And there's a lot of passion in this field, and you gotta love it, but it makes it tough sometimes to compare results and get some consensus. So a lot of the evidence in animals that just live up at high altitude suggests that they have a greater oxidative capacity. And that was actually the idea for a long time before we started looking at humans and climbers, that generally that's what happens. You just get better at using oxygen up at high altitude. But more recently, human studies that have started to look at this show either not really any change or even a decrease in oxidative capacity. And that's when they're measuring, things like what we do now with these machines that can actually, in situ, in vitro with a small piece of muscle, see how well it can oxidize different fuels. And there wasn't a lot of evidence for a lot of improvement, but there's a little more detail I could get into here, but generally not a whole lot happening. The other thing that's sort of a problem in the field is you can't study everything every time you do a study. So very often conclusions are based on marker enzymes. And one enzyme that we all might agree is a very good marker for a process might not be. Metabolism is not driven by one enzyme, even the rate-limiting enzymes. And they can be allosterically regulated. So the amounts of one or two things might not actually reflect what's happening. And then of course we pick different things, and so that makes it even tougher. Throw into that the variations that we might have in genetic background or training history, climbers versus non-climbers, and we're studying muscle metabolism. Obviously, those things are going to make a big difference too. So there's a lot of variation. The general consensus has been for a long time, ever since, and maybe before this, but for me, it looks like the landmark study was Brooks' study from '96. It showed that up at 4,300 meters, Pikes Peak type altitude, for a couple of weeks, that there was this increase in glucose uptake and what appeared to be less use and uptake of fat, or at least less use of fat. And so a shift to glucose. And this made a lot of sense. And very often, if you know your biochemistry, and this is a slide from one of my classes that I don't even hold my students responsible for, so I'm not gonna go over in detail. But if you do all the math and you see how much ATP you get per oxygen from glucose, or the same amount of carbons from fatty acid, you get more ATP per oxygen from glucose. And so that means it's like a more oxygen efficient fuel. And obviously everything in biology that it makes sense, right? So if we're going to go up to altitude and use more glucose, it must be because our bodies just know that we're going to get more energy from that. So when there's less oxygen, let's just use glucose. And there's a little more basis to it than that, but this has been cited very often as the logical a basis for why we'd shift to glucose at high altitude. And of course, this is just because fat generates a bunch more, not a bunch, actually, just in beta oxidation, more electrons removed per CO2 produced than carbohydrate. And so we have to pick those up with oxygen. And so you end up consuming more O2 than CO2 is produced, and compared to carbohydrates, compared to the same amount of carbons. It's really just the beta oxidation cycle versus pyruvate dehydrogenase, if you know your pathways. So this is actually pretty convenient that we know this, because we can then measure the amount of CO2 we're expiring, the amount of O2 that we've consumed per breath, usually a metabolic cart, which I know a lot of you are familiar with, and then get that ratio and get a sense for how much fat versus pyruvate or carbohydrate we're oxidizing. That's the respiratory exchange ratio. The minimum being 0.7 if we're at the tissue level at least, and maximum at the tissue level 1.0. And of course they can go beyond that, but that's the range. So the other kind of justification that's thrown in often to explain why we would have this shift from fat to carbohydrate use at high altitude is the hypoxia-inducible factor alpha, or HIF1 alpha, the master regulator of hypoxic adaptation. And this has been a really hot area for a long time, especially recently in the cancer literature. Semenza, Radcliffe, and Kalin, I think, got the Nobel Prize last year, 2019 or 2020. I think it was 2019 for this work, and it's really is a pretty fascinating transcriptional regulator that is stabilized. The protein is stabilized when oxygen is low in cells, and it does much what we're saying. It inhibits pyruvate oxidation, increases glucose uptake for essentially glycolysis, triggers things like mitophagy, or the destruction of mitochondria, inhibits mitochondria, can change the cytochrome oxidase subunits to make them more efficient. So you have a little less mitochondria, but the ones you have might be more able to use the low O2. And so this happens in cells. It's well established. It's particularly relevant in cancer cells now, where you have these tumors that are very thick and don't have blood supply, and the HIF1 alpha signaling is probably critical for how they're surviving. It probably is induced in ischemia, obviously any kind of time you have no O2 supply. But the question really is, are skeletal muscle really getting hypoxic enough? That is, the PO2 inside muscle, is it getting low enough at altitude to have this all turn on like this. And there's actually evidence that it doesn't, at least at rest, showing that oxygen extraction at, this is simulated 5260 meters, up around 17,000 feet, that the uptake of or extraction of oxygen by muscle from arterial blood is about the same as it is at sea level. Now start exercising, and of course you're going to create a problem. But if you exercise intensely anywhere, you might have that issue where you could consume, at least transiently, all the oxygen that's in that cell. So the question really is the muscle sitting in a chair up at 18,000 feet, is that muscle really hypoxic? And I'll leave that as an open question for now. Get to it. So, okay, so if we go to altitude, we don't have enough oxygen, so we want to use our more efficient glucose. We have this HIF activation, perhaps, that shifts us to glucose from fat. But there's evidence out there, even earlier, actually, for actual more fat utilization. And it's not as popular, and there's not as much of it out there as the glucose story, but there's some. And one particularly young, astute researcher found the same thing, more fat utilization in women up at this 4,300 meter, which was in contrast to the studies shown by Brooks in men, that there was a shift toward carbohydrate utilization. This is actually an RER graph over time showing rest and exercise, where at 4,300 meters, you can see the RER is actually lower than it was at sea level in these women. And I hope, Barry, I'm doing this enough justice with what I've said. But I love seeing this, because there just wasn't a lot out there. I'll give a little preview. We saw the exact same RER in our subjects at high altitude in our study, men and women. But suffice to say, it's not unheard of that fat utilization in humans could also be increased at altitude. So maybe it's both. And I'll tell you, and already it is. It seems like glucose uptake is up and fat uptake is up, and oxidation. At least that's our study. So how could we both be right? If they seem so opposite in the way that we about them, and hopefully I'll explain that. There's also some evidence from these native high altitude dwelling humans, so first generation Tibetans that are born living up at very high altitudes all the time, seem to have these greater expression of fatty acid oxidation enzymes. This is just one paper, and I had to find papers when I was starting to look for this fat story, and this was one of them, one of the few that showed that even in kind of chronically, not just sea level dwelling people going up to altitude for a few weeks, but people that have been there for their lifetime, there's some evidence that there might be increased fat oxidation. And that's really, again, marker enzymes here, so who knows? But I think there's no error bars. It wasn't the greatest study, but there was some thought that this is possible. So it was at the time of the study, 2012. No one had really done a very thorough, or at least omics level, comprehensive analysis of how humans respond to high altitude. The name AltitudeOmics says it all. That was coined by Rob Roach, the PI on this study. And it was in collaboration with some other investigators at University of Oregon, and Andrew Lovering is the big one right here with Rob. And these guys really, along with I guess 25 scientists from 6 countries, and that's including us, with a team from University of Oregon and a postdoc or a Ph.D. student I had here from CSU, went down to pretty much get as much information we could get from people that are going to be up at high altitude. This is in Bolivia, 5,200 meters, about 17,000 feet, for up to 16 days. And this is the altitude profile that they took the subjects up. And we didn't do the entire study. We only got muscle biopsies at sea level and at altitude after 16 days, ish, a week and a half, really, because they came down and back up. But generally sustained for over a week up there at 16 or 17,000 feet. So these are when we got our samples. So we were brought into the study pretty late. I wasn't part of any of the design or even actually go. I didn't even go. My student, Catherine Lay, went and got all this work done. It was not easy, physically or logistically. But there's a lab up on top of Mount Chocotaya, and they were able to collect these samples and do this kind of work that I'll explain here. So we had 7 males and females that we were able to get samples on, all young, all students really from the University of Oregon. And we got them again at baseline where they were in Eugene, and then again after 16 days up on top of this mountain in Bolivia. This is Hans Dreyer from, he's a professor now at the University of Oregon. This is actually Eric Niger, the CEO of the Ouroboros Instruments company that makes the respirometers that I think you guys have and we have. And this is my student in the back, Catherine Lay. So they were able to get pieces, of course, from the vastus lateralis that we could freeze for proteomics and metabolomics, and then freshly tease apart individual cells, largely our individualized cells, into these bundles that we can put into the respirometers to measure oxidative capacity. So that's what we did. This stuff was frozen, and we tried to get that back to the States without thawing, and that was successful. And then ran these at CSU, and all this mitochondrial analysis was done in that lab when the biopsies were taken. So the general characteristics of the subjects after 16 days were kind of what you expect. Everybody had lost some weight up there. The O2 sat and arterial oxygen content was lower and went down and stayed lower. May have recovered a little bit, but stayed lower than sea level values even after 16 days. But you see these these acclimatization effects that you'd expect over 2 weeks. Here's the data on the RER. It was actually quite high at sea level, which might be sort of a problem here, but it went down and even upon first arrival, and then even further after 16 days. So similar to some of the data I mentioned. And that, of course, suggests some kind of shift away from carbohydrate oxidation toward either fat or protein. But that it really isn't that's really, it's just the RER. So we would need to do more to know that, and we did. VO2 max, of course, is lower, recovers a little bit, but not statistically significant. So generally expected a response to both acute and 16 days up at high altitude. When we did the metabolome, how do you present this stuff, right? That's one of the great challenges of this generation of science now with bioinformatics and omics. How do you capture 10,000 data points? And I didn't have that many. I had just a few hundred, but this is one of the ways we did. And essentially what it is, it's just a log2 scale and a pie chart showing these different categories of metabolites. And I chose this kind of format to just illustrate that everything was up. Or not everything, a lot of stuff was up. Amino acids, almost every single one was up. Carbohydrate metabolism, same thing. Nucleotide metabolism, which I didn't even think to look at, but there it is, was elevated. The Tca cycle was kind of interesting, and I'll get to that, but some were up, some were down. Fat metabolism, mostly up, but not always statistically significant, a whole lot of variability. And then you see ketones were actually down. It was the only thing that looked to be pretty consistently down. But overall, there was a lot of accumulation of metabolites, and so mobilization of fuel within the muscle viscous. I don't have the similar graph for the serum, but I have some key pieces that I'll show you. So in the muscle, a lot of metabolites are sort of up or increased relative to sea level when you've been up at altitude for 2 weeks. So more fuel is available, and that makes sense. If we've got this, cortisol, we mentioned, is going to trigger things like catabolism that could explain the increase in amino acids. If you have some adrenal drive, maybe it's still present even at 16 days. You're going to have mobilization of intracellular glycogen and maybe uptake of glucose. Fatty acids could be mobilized or uptaken. So there's a lot of fuel in the muscle. That's the point. The other thing that was interesting and unexpected, but we were able to get the ATP to ADP ratio, or so ATP and ADP and phosphate and AMP, and you can see them here. And this is a log scale again. So, but you can see there was a statistically significant and this is maybe not as pretty as it could be presented. You can see there's some variability across subjects, but there was definitely an increase that was significant in ATP and a decrease in ADP. Phosphocreatine was higher, creatine was higher, but the ratio of the two was not different. So what is supporting this higher ATP to ADP ratio? This is all at rest. Remember, these subjects were not exercising. They were allowed to walk around, but nobody was climbing. They weren't athletes that had to get their workout in every morning. They were just told to walk around the camp, take pictures, chill, play games. And that's really, I think, what made this study pretty unique. Another study was done a couple of years ago at Everest by the group at Cambridge, and they've done it a couple times. And they trek to base camp, and that's the control group. And then there are subjects that will climb, and they have carrying large loads. So it's a little different when you have all that kind of work. So this is just sitting around. You have a higher ATP-ADP ratio. Lucky for us, this even with the Everest group, this is a previous study that did in vivo measurement of high energy phosphates and found the same kind of thing, that there was lower ADP, higher ATP-ADP ratio, no change in phosphocreatine, but a same kind of increased what I called phosphorylation potential or greater ATP relative to ADP. Generally, these things are pretty constant if you're rest. You exercise, ATP will fall, ADP will climb. That'll drive the bioenergetics of the muscle, like I talked about earlier. So why would it be higher? And this was kind of an interesting question that we had, and I've had to think about for a long time. Most of the ATP, of course, is going to be made inside mitochondria. Phosphate is available. It was higher, as you can see here. There's this creatine kinase that's in the intermembrane space of the mitochondria that you might not know about, not like the one that's in myofibrils, that is there really just to ensure that ADP is always applied to the ATP synthase. So it essentially can hydrolyze the ATP immediately after being produced, and then harnesses that energy to phosphorylate creatine. And then you have like a phosphorylation cascade or phosphotransduction cascade that can go and essentially supply that energy to other parts of the cell. And so this, without showing you all the proteomics data, this protein tended to be higher, right? It was higher after our statistical analysis. So maybe that's part of it. And phosphocreatine is higher, so maybe it's this phosphocreatine system that was enabling higher ATP levels. Another pathway that can maybe influence this is the pre-nucleotide cycle. That was, and the only reason I'm showing you this is because all those proteins were elevated. And I didn't even know really what this thing did when I first got this data, as the joy of omics. And it interacts with the malate aspartate shuttle to basically get reducing equivalents into the mitochondria. But really, its major function, aside from degrading nucleotides, which apparently was happening, uric acid, hypoxanthine, these products of adenosine breakdown were elevated. That actually can increase ROS production. This reaction here produces H2O2, and so this could be a source of ROS at high altitude. But the real point of this pathway in muscle is to generate ammonia, which helps produce glutamine, which is actually excreted or released from the muscle cell, and ultimately goes to the kidney to assist with like acid-base balance. So this is a major helper when you have protein catabolism, which we know is up. So maybe that's it. And I don't know how that would influence the ATP-ADP ratio, but maybe that was part of it. This increased ATP-ADP ratio we know can influence glycolysis, or if you don't know, I'll explain. There are some rate-limiting steps that are sensitive to this ratio. And so I'm going to take a look at that in a second. But first, I want to show some of the data that we had from the barometer. And whoops, sorry about that. If you look at just the total respiratory capacity of the muscle, that is, you take a piece, you give it all kinds of fuel. So we give it pyruvate, so a carbohydrate, glutamate, an amino acid, and palmitoyl-carnitine with malate, a fatty acid, and a TCA intermediate that can go straight in. Nothing was really different between sea level and the 16 days at altitude. And I did this under what we call uncoupled conditions. That is, we throw in an uncoupling agent that prevents formation of a proton gradient. So you get to see how fast the whole thing can run. So the capacity of the muscle to just oxidize fuels was not changed by high altitude. If you don't put the uncoupler in and drive the same process only with ADP, you get this sort of variable trend, not really reaching 0.05 significance for an increase. But if you take the two and look at how much of this total capacity is being accessed essentially by this ADP-dependent respiration. Really, the ADP control, in a sense, of respiration. It was actually pretty interesting. There are a couple trend buckers here, but most significantly increased. This is the ratio of the oxphos links to total capacity. Was highly significant. So this suggests that something in this mix of substrates, when ADP is present, is able to run higher, and it's an ADP sensitive thing. So the oxphos capacity is higher, not just the enzyme, the respiratory capacity. So maybe that is some kind of a reflection of how we can have more ATP relative to ADP, but it's not really clear. But this was an interesting finding. And that was, again, with all these substrates present. So if we look specifically at glucose or carbohydrates, we see the same kinds of data that others have shown. Glucose was lower in the serum, higher in the muscle, so perhaps more taken up. Lactate was higher in both compartments, particularly muscle, which is interesting because you would expect it to be driving it out, but both are significant. And these are ratios too, by the way, so these aren't absolute values. So this is just relative expression of that metabolite. Glucose 6-phosphate, that first intermediate when you bring glucose into a muscle, these are both muscle data, was higher. Although again, you see not everybody went up. If you look at this capacity to oxidize pyruvate, it actually came out to be statistically significant, but this is very unimpressive, and it's certainly not lower. So let's just say, pyruvate oxidation capacity isn't suppressed. Maybe it's a little bit higher, but obviously quite variable. So is this evidence for more glycolysis, right? There's more glucose around. We're not maybe oxidizing too much more pyruvate. We have a lot more glucose that we phosphorylated, maybe in some, at least, individuals. Lactate is up. That just sounds a lot like glycolysis to me. And of course, it is. But glycolysis isn't just this one line of reactions that produces ATP. And I think a lot of us know this, but maybe we don't think about it that much. First of all, glycogen, breakdown and storage enzymes are all up. So obviously the muscle is handling a lot of glucose and glycogen. Which direction it goes, who knows, but the capacity to store and break down glycogen, at least by protein expression, was elevated. But if the ATP-ADP ratio is up, and that's what red means here, by the way, then we know that that's going to, first of all, drive hexokinase, which is a kinase reaction that requires ATP. And we know we have more, at least a tendency for more glucose 6-phosphate. We keep going down the pathway. Don't worry about all the enzymes, but the gray ones were not up. The red ones are. And we get to the middle of the pathway. We know that this PFK enzyme, phosphofructokinase, is sensitive to ATP-ADP ratio also. It's actually inhibited. So would we actually go through here? I don't know. We can go another route. The pentose phosphate pathway comes off this as well. And if you looked at the enzymes in there, and the not the enzymes, the intermediates, the metabolites were elevated. And ribose is one of them. This is where we make pentose, so 5-carbon sugars, which can go into nucleotide synthesis. And perhaps this could, combat the degradation of purines purine nucleotides that we saw in the couple previous slides. But this was elevated also. It can feed back down into the glycolytic pathway. And in both cases where it feeds back in, that metabolite was elevated. It's getting heavy now, I know, but if we keep going down the glycolytic pathway over to pyruvate, we see some of the downstream enzymes were elevated. And especially immediately following this, the GAPDH step. And this was a new one to me too, but pyruvate was lower, lactate was up. Okay, maybe this is just running all to lactate, and we definitely saw an increase in lactate. But there's another pathway that can be driven by inhibition of pyruvate kinase, which tended to be lower in the muscle, and we know is inhibited by ATP and alanine, which are both elevated in the muscle. And this other pathway is called 1-carbon metabolism, for lack of a much busier term, but it's essentially the folate and methionine cycles that generate a whole lot of different important molecules, among them creatine and purines. And so these, I have a slide on this. It's actually got a supplemental in the paper, but it's so busy, and there are so many metabolites, and to go through them all is just we're going to run out of time anyway. But suffice to say, this pathway seemed to be heavily fueled. Increased in rate, we don't know. These are all static measurements, but there were more metabolites around, and among them was creatine. And so this is maybe some circular logic here. I don't know where it starts, but more creatine seems to be supported by this kind of redirection of glucose. So the reason for me to share all this stuff with you is to impress upon you that just because you're taking up more glucose into muscle and you're using more in the glycolytic pathway doesn't mean it's all about just getting some ATP out of it before you get to lactate. There are a lot of more fates of glucose than just pyruvate and just ATP producing steps. And these seem to be elevated at high altitude. Pentose, phosphate, and 1-carbon metabolism being two big ones. Of course, pyruvate can enter into the mitochondria. It's lower, perhaps not even going to be entering there, even though we know that in vitro the muscle fibers are capable of oxidizing pyruvate. In vivo, they might not be much if this ADP or ATP to ADP ratio is inhibiting it. We don't know. I'm going to skip ahead just for time. Let me show you now for a little sidestep here. I'll get back to the glucose story in a second, but the muscle accumulation of free amino acids was also pretty profound. Virtually every single one except cysteine was elevated, particularly aspartate and your branched chain amino acids, your leucine and isoleucine, with the three top winners. Serum alanine was elevated in the serum. Glutamine trended that way, didn't make it entirely, and then you have these markers of kidney processing, perhaps, or protein catabolism, urea and creatinine elevated. Succinate was up. I'm not going to get to that, but that's an really interesting one. Why succinate gets ejected from mitochondria at high altitude. But what was really interesting is the Tca cycle. You think of it as just this thing, the Krebs cycle that we run to get our NADHs to fuel the respiratory chain. It was generally lower. If you looked at the first kind of front end, if you will, citrate wasn't different, but the next three substrates were lower. But then malate was pretty significantly elevated. And so what's the deal, right? I mean, I thought these were all kind of in the same program. Why would like the back end be up, or why would malate in particular be up? And I'm going to take just a minute here. I guess I'm running a little late, even if we consider the late start. But and I always hesitate to present this kind of slide, but I'm gonna go for it. See, hopefully you can follow. And my apologies if it's just too much. But we know that glucose is elevated. We know that there's these side pathways that are maybe favored, that are elevated, perhaps due to the accumulation of alanine and ATP-ADP ratio elevation. And pyruvate's lower. We think that maybe that could be because it's being converted to lactate, but LDH was not elevated. So what's driving pyruvate out to lactate? Here's the Tca cycle. You can see succinate and alpha-ketoglutarate were lower. Some of these enzymes trended higher, but again, malate was much higher, and these intermediates kind of, as you get to malate or preceding malate were lower. So why would malate be higher? Let me remind you here: when you have the Tca cycle, these don't have to stay in the cycle. These intermediates are able to be removed, cataplerosis, or come in from other pathways, anaplerosis, and that's something that happens when we have a lot of free amino acids around. So particularly leucine and isoleucine, these branched chain amino acids, when they're oxidized, can be converted into keto acids that will feed back into the Tca, or converted to glutamate if we use alpha-ketoglutarate. And muscle, if it has a lot of this, and we know that that's happening at high altitude, uses glutamate to make glutamine in order to eject some of this this nitrogen. And again, if you remember the pre-nucleotide cycle, it uses aspartate in order to generate the ammonia to synthesize glutamine. And that's kind of its big role when there's a lot of proteolysis. And that was elevated. So what we're doing essentially here when we have a lot of proteolysis is robbing the Tca cycle of alpha-ketoglutarate in order to get rid of our nitrogens. And the other amino acids are going to feed in different places, and it just gets too much. But suffice to say, this is a large way that we're getting rid of our nitrogens. And really the muscle can just release glutamine and alanine to get rid of. It can't just get rid of all of its amino acids if there's excessive amounts. And so glutamine is a big one. And you can see here this inverse relationship between the two. So essentially, glutamine is being generated by glutamate largely until they both start to accumulate. Fumarate is produced as a byproduct, and that gets converted to malate. So now we have some more malate. So maybe things are starting to make a little more sense on this end. That arrow and see my thing here. Alanine is produced by sort of competing with glutamate here for glutamate by the alanine transaminase to form alanine from pyruvate. So here's another fate of pyruvate. Alanine was up, and alanine can be ejected. And this is another way that the cell can kind of get rid of nitrogen. So it's a whole other pathway. The malate aspartate shuttle can try to keep this going essentially by regenerating glutamate from alpha-ketoglutarate. And if it does that, it's going to form more malate. And this essentially shows, you have 3 arrows going to malate, probably how malate starts to accumulate, both in mitochondria and in the cytosol, probably more in the cytosol. And there's an enzyme that was elevated. One of the strongest increases that we saw was malic enzyme, which I hadn't really even thought about. But malic enzyme converts malate to pyruvate. And maybe malate accumulation is what's actually driving a lot of this lactate production through this linear pathway here. And if you actually look at the correlation between malate and lactate, it was essentially a 1. And so maybe at the very least, malate accumulation is maintaining pyruvate for all of these fates that it might have. TCA might be inhibited, so really maybe just alanine synthesis and lactate synthesis, but not necessarily more glycolysis. And that's the real point of this slide, as busy as it is maybe all this lactate production, which is, sometimes it's called a paradox at high altitude. Even you'll have elevated lactate. And even when you've acclimatized, there'll be lactate present. Is lactate a product of glycolysis always? If we're using it as a marker of glycolytic flux, the two seem to disconnect sometimes. And at high altitude, they seem to disconnect. And maybe it's because there are other ways to drive this lactate production besides just glycolysis, because we can get pyruvate from malate, and that seemed to happen here, perhaps. All right, just a quick review, and I'm going to start to close here in a couple of minutes, but we have this increase in coupling control, perhaps, that is leading to a greater ATP to ADP ratio. Maybe both happen. Whether they're related, we don't really know, but those were 2 interesting findings that suggest there's this greater capacity for ATP levels to stay high, perhaps to allosterically regulate metabolism. We don't know, but that seemed to happen. And the catabolism of protein creates an interesting shift or rewiring, if you will, of substrate flux through the Tca cycle that probably has to do with trying to get rid of nitrogens from the system, but also ties into these pathways above, like the Pre-Nucleotide Cycle. And how this could all be coordinated isn't really clear, but this seems to be at least the way that I tried to put it together as a way that things could be working. So the question remains, if you've got all of this happening, where is the ATP coming from? And what is the actual mitochondrial ATP contribution here if it's not pyruvate, if pyruvate oxidation is significantly elevated? And so where I've been leading here with the fat story is that the fatty acid oxphos, fatty acid linked oxphos capacity, that is, you put little muscle fibers in our machine, give them fatty acids, add ADP, they're able to oxidize at a higher rate at a high altitude. Conveniently for us, there was a nice correlation, not perfect, but what can you do? This is the respiratory exchange ratio and the fatty acid oxphos rate from the fibers we've actually took from the biopsy. That's a pretty decent correlation, as you'd expect, right? As there's higher oxphos capacity of the tissue, the RER in those individuals was lower. So that suggests that maybe this is actually a legit measurement, right? Ripping a little piece of muscle, throwing it in with some fat, seeing how much it can oxidize. Maybe that actually is a way of assessing how much fat that person can oxidize. Things like leak weren't different, but coupling control was, again, suggesting that maybe it was fatty acid that was carrying that effect of having a greater ADP-linked respiration, or energy demand-driven respiration when fat was in that mix, because pyruvate and succinate coupling, that is how well they're driven by ATP, were not at all. So it was really fat in that mix, maybe, that I showed you initially, that was carrying that effect of having a greater ADP-driven respiratory capacity in the muscle that was independent of just the total enzyme capacities, all told. So maybe there's a selective improvement in long-chain acetylcarnitine oxidation capacity. I'm going to whip through this really fast, folks. I'm out of time, but suffice to say, we did a ridiculous amount of analysis of all the proteins in this pathway. Interestingly, there's a whole system for oxidizing long-chain fats on the inner membrane that most of us aren't told about. The beta oxidation cycle that we hear most about is actually in the matrix. And there are these long-chain specific enzymes, very long-chain acyl-CoA dehydrogenase, and trifunctional protein, which actually has all three remaining enzyme capacities for the beta oxidation cycle. So these two are basically a long-chain beta oxidation cycle bound to the inner membrane, and they were elevated, or at least they were relative to everything else trended up. These are relative to citrate synthase elevations. So that goes along with the fact that maybe palmitoylcarnitine oxidation is increased simply because there's more machinery there to do it. Of course, why ADP was driving it is still an open question, but that's all I'm going to say there. To wrap up, I didn't want to ignore HIF1. In fact, we couldn't. This was something that a reviewer asked for. So I guess I did want to ignore it, but I couldn't. HIF1 alpha was not present in any sample, either at sea level or at high altitude. So this is that master regulator that's supposed to go up with hypoxia. We can actually find it. This was all Hans Dreyer at University of Oregon. He actually does pretty interesting work where they do tourniquets. They study muscle pre and post tourniquet application during total knee replacements. So this makes the muscle very ischemic during the total knee replacement, and then they release it, and then they study how the muscle responds to that kind of ischemia reperfusion process, if you will. And they were able to see some trends. I don't know if you can see it here. I can barely see it on mine, but with some ischemia and reperfusion, there was some HIF stabilization. If you take some muscle cells and culture them, you'll see it even in the presence of oxygen, and it goes up a little bit when you take the O2 away. But what the point here is that our antibody was working. It is possible to get a HIF1 alpha stabilization in human skeletal muscle, but sitting around at 17,000 feet doesn't seem to do it, at least after 16 days. Maybe we missed it. Maybe it was stabilized the first day. A lot of this transcriptional stuff was set in place, but by 16 days, all the stuff I was talking about was happening, and there was no HIF1 to be found. So we don't think that that's at least the major driver at this point, at least after 16 days. There are lots of other players. AMPK has been thought to play a role here. If there's energetic stress, of course, we don't really have evidence for that. There was high ATP-Adp ratio, and there was no change in AMPK content or phosphorylation status. So we looked just to be complete. OPA1 is another one. This was another one a reviewer asked for that's been shown with hypoxia to alter things like fission, fusion, mitochondrial dynamics. And we looked at multiple weights of this because it can be truncated in different lengths of this protein, and no differences there. What we did find, which was pretty interesting, and again, I went after this because we saw this change in fatty acids, but the peroxisome peripheral activated receptor, PPAR alpha, is a master regulator of fat oxidation and is driven by fatty acids. So fatty acids can actually bind the transcription factor to activate it. And so then it goes to the PPAR responsive element in DNA and ultimately increases expression of enzymes for the transport and oxidation of fats. And we blotted for this, which isn't the best thing, right? Because we don't know if it's going to DNA. We didn't, look at any transcripts. But what was interesting is it came out as at 2 weights. 50 is kind of the standard. We also saw bands down at 27. If you're not familiar with Western blot, you basically just run proteins out and then bind an antibody that you hope is specific to your protein, and then look and see how much is there relative to other groups. So this is very consistent. Here's just 4 individuals, but you can see that these lower molecular weight bands were higher in the at L16 compared to sea level. And this is these are the same individuals, so 2 individuals actually, pre and post altitude. And so we had a lot of statistical power, and we were actually able to get statistical significance with this. It's obviously not super strong, but what was cool is if you correlate these levels, again, these are just like density ratios, relative to total protein Ponzo staining here. You correlate that PPAR alpha expression change with the oxphos capacity driven by fatty acids, it correlated, which again, the individuals that had higher PPAR alpha expression had higher rates of fat oxidation in their muscle, at least in vitro. And of course, I'll remind you, the RER was lower intended in everybody to be lower. And so perhaps this is it. I mean, maybe PPAR alpha, because of all the fatty acids that are mobilized or activated, and this actually drives fat oxidation. And it doesn't make terrible sense. I mean, it makes sense. It's not terribly controversial if you can just give up the fact that there's truly a lot of hypoxia around to activate HIF to prevent mitochondria from doing anything. So I'm going to stop very soon, I promise. I wanted to get to this because it's what we're doing now. We have this hypothesis. You have to ask the question, like, great, Chicco, we love when you give talks, you just get all excited about all your data. Does this matter at all? What are the metabolic, kind of implications of this? Is it good or bad that all these things are happening? And, since the amino acid mobilization or proteolysis seems to be such a big part of the story, we had this hypothesis that glucocorticoids, or cortisol in humans in particular, what if that's driving a lot of this metabolic remodeling? All those amino acids that are flooded that have to rewire the Tca cycle. What if we just blocked that pathway? Would that prevent acclimatization? Or what if we just turn it on by adding something, like giving someone a glucocorticoid like a dexamethasone? Would that induce some kind of acclimatization? But we don't really know, but we asked the question. And of course, physical performance is something we want to look at. So I have a new student, and a lot of you might know him. He was, I think, an outstanding undergraduate in your department as of a couple of years ago. Now he's an outstanding and exhausted graduate student in my lab. This is actually from a couple hours ago. This is our 5th day in a row of about 18 hours. These are long days getting all these experiments run, taking down 2 rats. But his thesis is looking at this glucocorticoid signaling in rats that are exposed to the same kind of altitude or hypobarrier, at least, that we got up in Bolivia. This is all garbled, so that sucks. But in the hyperbaric chamber over in our building in physiology. What this mess here is saying is we're giving them essentially the same kind of time at high altitude that the humans had, about 16 days up at the same altitude equivalent, right? Simulated 17,000. And we're either giving them dexamethasone during that time, inhibiting glucocorticoid signaling with an inhibitor called RU46 or mifepristone. They're all male rats because mifepristone can also hit progesterone receptors. And then we're also doing a group where we're just injecting with dexamethasone prior to exposure to high altitude to see if we can sort of prepare them or condition them simply with proteolysis signal or whatever we would say dexamethasone would do for high altitude. And we don't have a lot of data yet, but this is, here's Luke running some rats. And what we're seeing is, first of all, dexamethasone, this has been shown before, seems to increase LV systolic function. And this is just at sea level, or normal oxygen. Despite the fact that the animals are losing what you'd expect, 15 to 20% of their body weight. This is IP injections now, about 0.75 mg per kg. It's a protocol that was established in rats to induce negative protein balance, and without being deadly. So these animals lose a lot of weight, they lose a lot of muscle. It looks like they're selectively losing white fibers when we're doing the the morphology and dissections post-mortem, but we don't have any data on that yet. The interesting thing is the animals that are up at high altitude, they run better at high altitude. Okay, so after 2 weeks up at high altitude, the animals do better on our graded exercise treadmill test that we run them on. The interesting thing is if we just inject rats with dexamethasone for 3 days or 8 days, 3, it's getting there. 8, it's almost exactly the same. So animals that just get dexamethasone, lose a bunch of weight, frankly look like crap. We pull them into the chamber, having not seen, they had been acclimated to the treadmill and normoxia, but they hadn't seen hypoxia. They go in and they run as well as animals that have been up there for 2 weeks. So that's really exciting. And we've got about 6 or 7 animals in here now, I think. So it's looking pretty strong. It's pretty tight. Animals that were getting dexamethasone in the water at a high altitude, and we only got 4 of these, I think, now. They are superstars. It's actually really interesting to watch them run. They're light on their feet. They're hopping, several minutes longer than some animals make it. Certainly when they started. And I don't have the pre-post data here, but suffice to say that this is pre and post we have on everyone. So normoxia versus hypoxia with dex, this was actually very similar. So the animals are, I won't say they're impervious to the hypoxia, but they're doing very well when they get there. So this bodes well for Luke's thesis and our hypothesis, and of course we're going to do a whole lot more analysis to try to see if all this fits. All right, my apologies for going over. I'm just going to finish now with take-home messages. If there's anything you really could take from this that's important, I think we need to be very careful about defining our terms. High altitude, hypoxia, hypoxemia, these might be mutually exclusive. That is, just because we're at high altitude doesn't mean we have hypoxia in tissue. We may have hypoxemia, but that doesn't mean we're going to have hypoxia in tissue. Some people who are acclimatized at high altitude might not even have much O2 sat desaturation. So what is hypoxemia? How low does this need to go to trigger the responses that we think are occurring? So I think this is a source of a lot of variability. The use of our terms and it's not faulting anyone. I mean, I've used these sloppy myself, probably even in this talk. But we need to be careful about what we mean and when we say these terms, because they're not all the same thing. The other thing is, just because a muscle is taking up a lot of glucose doesn't mean it's relying more heavily on glycolysis or glucose oxidation. There are a lot more effects of glucose that are relevant in skeletal muscle. This is really being shown in cancer nowadays as the metabolic revolution in that field, it seems. All the places that glucose is used for anabolic pathways. Nucleotide synthesis, ribose and purine synthesis through the pentose, phosphate, and one-carbon metabolism pathways. The other thing, relying on marker proteins or single metabolites, even single indices of oxphos function might mislead you. If you don't, we can't do everything all the time, but very often if we just pick one of our favorite markers, it might lead us in directions that are incorrect. I didn't note this in one of my crazy fatty acid slide, but the 2 or 3 fatty acid enzymes that are most commonly used to be markers of fat oxidation capacity, those were not elevated in our subjects. You heard a lot about all the stuff that was elevated, but those marker enzymes, beta HAD was one, CPT1 was another one. They weren't up in these subjects either. So if we had just stopped there, we would have concluded, like everybody, that there's no change or increase in fat oxidation. But there clearly was, given all the other measurements. And finally, protein degradation and the sympathoadrenal drive that may be behind that, cortisol perhaps in particular, may be primary drivers of this adaptation to high altitude, and maybe other what we call hypoxia-related pathologies. COPD, heart failure, also conditions where there's a lot of sympathoadrenal drive. We see hypoxemia and we think maybe it's HIF-driven or hypoxia-driven, but in many cases, maybe the catabolic state is really what is driving a lot of the changes in the muscle, not low O2 per se. With that, I will finally stop. I don't know how far over I am given the late start, but for sure over. So I appreciate anybody who's still on the line. I can't see. Maybe I'm not just talking to my computer alone. I don't know. But I have to thank Rob Roach, who's down at the Altitude Research Center in Denver, Anschutz, and his team who brought me in on this project. Here's the team that did the muscle work with Eric Niger, who supplied the machines and the expertise. Really, I was new to all this when this started. And Catherine Lay was really a superstar student that led all this and had lots of help. Hans Dreyer in particular, who took all the biopsies and was our muscle man. Here's Eric, and the team at Ouroboros was really a big investor in this and has been in my lab and a lot of my work throughout the years. And so it's a really great company, and he's a great guy, and a lot of the work they do to harmonize research in this field across labs and countries is really remarkable. It's really been fun working with them over the years. We had all the metabolomics done down at Denver. Proteomics was done up here, and that they were both fantastic. And this was all funded by DoD, mainly through grants to groups in Oregon and Rob down in Denver. So with that, I'll take any questions if there's anybody left and has the patience. And thanks again for the opportunity to share some of this. I hope it was interesting. I'll stop now. I'll stop sharing.
All right, thank you, Adam, very much. So we'll take a question or two, and then I think we'll wrap up. We're a little bit over. So I see Barry had a question in the chat. So Barry, did you want to unmute and ask your question or phrase it however you'd like?
Sure, I can do that. But Adam sort of answered it. And Adam, I'm glad that your newer data, match up with our, at this point, Matt Hickey would call classic data, what I would call old data. But and I really agree that sympathoadrenal response, glucocorticoids causing that proteolysis is really a big driver of what we call altitude acclimatization. So the question was, you almost unavoidably also have an energy deficit that goes along with that. Is that also, contributing to some of the outcomes that you see? And is that just unavoidable? Is that just part of the altitude acclimatization response?
Yeah, great question. And and I even in the paper before we started doing these running studies, when you short circuit the TCA, is that going to limit your oxidative capacity and lead to a energy deficit, as you say, in a good way? So you almost have evolved a system to handle all this degradation, become, efficiency is a term we all reach for, but, like do less with less, and then ultimately have less to support. If you can get rid of the muscle, have less fuel so you can't work as much, and then, this may be what's happening. But then, we inject these animals with dexamethasone. They look terrible. They're terribly glucose intolerant. So there's just more to it than just the dexamethasone, because altitude increases insulin sensitivity. But these animals look terrible, but then they run really well. So I was surprised at that, actually. I mean, the hypothesis could have gone either way, but is it? So they're running better, but it's very slow. The hypoxic treadmill test, they get up to about 20 meters per minute at the most. So they're not out there cranking it like they would at the output is very low. It's just they're able to do it longer than an unacclimatized animal, as well as an animal that's been acclimated. So the absolute level of performance is very low, but relatively speaking, they're doing better. And I think that's how I see it. Is you ultimately you're going to take a hit. Maybe this is to survive. Reduce the cost, give you less fuel to do things so that you can preserve it. But at the end of the day, you do better, or at the end of the week or 2 weeks, whatever it is for the adaptation, in that environment. Take that animal and run them on and we haven't done this, and maybe we should, but take those animals that do well at the Wimpy test at high altitude. They might be terrible, down at sea level, because they've lost all of this, muscle mass, and now they have to run at 30 meters a minute, and they can't even get up that fast. So I don't know the answer, but I think the efficiency or to degrade the muscle to cost less, have less energy, to do less. It makes a lot of sense from an evolutionary standpoint. But then when you get to a new baseline, you're now well suited for that environment, relatively speaking. If that makes sense.
No, thanks. I agree with almost all of that.
I'd love to hear about the other parts sometime. Thanks, Barry.
All right, we got a question from Chris Bell, and I think we'll go ahead and wrap it up. Chris, did you want to unmute yourself and ask the question, or you want me to just read it?
Oh, I want to hear it, Chris.
I don't know if Chris has a microphone.
Oh, okay. Somebody even lower tech than me.
Okay,
I miss Chris.
I will ask your question, Chris. All right. In your muscle serum slide, you showed greater ejection of lactate, alanine, and glutamine. All three of these can be used to create glucose through the Cori cycle, alanine glucose, and glutamine glucose cycles. Could this contribute to the greater reliance on carbohydrate?
Yeah, great, absolutely. I wouldn't argue against any of that kind of evidence that's been out there for a while, Chris. The only caveat I put to it is greater glucose utilization by muscle might not be just because we need it for glycolytic ATP production because there's not enough oxygen to fuel mitochondria. I think that's the big thing that I got. And of course, I love mitochondria, and maybe I'm a little biased, but the metabolome really suggests that there's definitely a lot of glucose available. It's in the muscle, and so maybe that is contributing. Maybe this the gluconeogenesis and Cori cycle, alanine cycle is contributing to all that glucose to come back to the muscle. And so to fuel these other pathways, in addition perhaps to just your good old fashioned glycolysis and glycolytic ATP production. But to expect that all that glucose is there to maintain the energy demands of a resting skeletal muscle just doesn't make any sense. I mean, under resting conditions, you're using fatty acids everywhere, even at 18,000 feet, I think. I mean, why wouldn't you? There's no huge energy demand. As I was just talking with Barry about, if anything, maybe it's a little lower as you start to lose the muscle. So the glucose that's coming in and being used and being recycled, like you asked, perhaps it's there to balance all this nucleotide degradation, to fuel, the one carbon metabolism cycles that are, who knows? There's when you start talking about the folate and methionine cycles, we could go on and on and on. Somebody could go on and on and on for a long time about what that might be doing. Creatine was the thing I pulled out, but it would make sense that you would redirect glucose to those, and it looks like that's what's happening. But yeah, for sure, I would say the liver helps out. Thanks for the question.
So one last little question from Chris Bell was a question about the Ouroboros working okay at a high altitude. I would assume you just kind of worked at a set PO2 and injected oxygen to the levels you wanted, or how did that work?
Exactly right. Yeah. So Eric was actually really interested in this. This is way back 2012, right? So they had never gone that high. So he flew out. He would have gone anyway, but he flew out to the builder of the machine to make sure it was working properly. Calibrated all this stuff. But yeah, as you said, Dan, yeah, the fibers require a high O2 environment to have stable respiration anyway, because of the diffusion limitation that results from permeabilizing them and having all the myoglobin come out. So you have to jack the O2s up to 300-400 micromolar to get stable respiration anyway. So and it's in a temperature controlled chamber at 37C. So all that is perfect and calibrated everywhere, no matter where you are, as long as the power stays on. So yeah, those conditions were identical in Eugene and in Bolivia. Good question.
That was awesome. All right, great. Thank you, Adam. So we'll go ahead and wrap up. Next week we have Marnie Bopart coming from the University of Illinois talking to us about integrins and muscle regeneration. So please join us for that. Thank you very much, Adam. If you could stick around with the 793 students, we'll do our after party.
Will do. Thanks, everybody. Apologies again for the long talk, but you probably expected it. I guess it's less of a problem, right, when it's virtual, because you can just click away.